And we're live right now, so I'm going to turn it over to Karen.
Hi, everyone. My name is Karen. This is going to be very awkward. I'm going to say this on the onset right now because I can actually not see myself. I do not know what I look like in this image because of some technical difficulties, but we're going to run the club as is. Anyway,
We'll warn you.
everyone.
We'll warn you if anything weird happens. We'll start to tell you if something goes horribly wrong. We'll be like, Karen, no.
All right. Welcome, everyone, to our well, it's not Google Plus anymore. It's now YouTube Live. So at any rate, welcome to our internet journal club for microbiology papers. And this is just a fun way for us to get together, or just us being any type of scientist, whether post in any point in our career. So I'm a PhD student, and Laura and Nicole will introduce themselves shortly, but just any point in our careers and get together and talk about these very interesting papers that are coming out. And the neat thing about this type of journal club is we don't actually know everything that the paper is all about. The point of this is so we can read a variety of different types of papers and try to get exposure to different types of microbiology. And so on the onset, for example, for this paper, there are a lot of complicated techniques that we'll try to walk through, but we'll discuss the techniques, see what we can take away from them, and try to come up with some, the point of our club is so that we can come up with neat ideas from these papers and gain a broad understanding of what microbiology is like today. And so on that note, I'll introduce myself. My name is Karen. I'm a PhD student at Simon Fraser University, and I am frantically trying to finish my thesis. And so I study a pathogen called Francisella tularensis and its virulence factors. So this is, oh no, I can't see Laura anymore. This is similarly up my, this paper is similarly up my alley, except for the bunch of microbiology things that we are go through, but there's a lot of neat techniques that I do understand here and I can appreciate it. And so I have. So that's it for me. I will turn it over to Laura.
Okay.
Yes.
Yeah, no problem. Yeah, I don't know if I flicked in and out there, but I could still hear you just fine. So I don't think you have to worry about that. So my name is Laura Williams. I'm an assistant professor at Providence College. Usually I have my little thing down there, but I got distracted, so it's not there. I might pop it up later on. And I think Karen just gave a really great introduction in terms of what the Journal Club is about. So this is our first Journal Club for the new academic year. Over the summer, we did a book club on books about microbiology for the public, which was a lot of fun and a really good excuse to read 3 great books. And so now we're transitioning back into our Journal Club. And as Karen said, you know, this is a great way to stay on top of some of the literature that I would definitely miss otherwise because everybody's busy. And it's also really neat because we have people from different stages of their careers and different parts of the world. So I'm actually outnumbered by Canadians, I think. So we got two people in Canada and 1 person from the US. So there we go. But we've had people join us from outside of North America as well. So if you're at all interested when you run across this in joining us, we welcome new people. We're always happy to have people join us. So do feel free to check out our website, which is linked in the video for this as well. And so then I will turn it over to Nicole.
Hey y'all, I'm Nicole Suktio and I tweet as Old Dusty on the Twitter. And I am a postdoctoral fellow at the University of Northern British Columbia in Prince George, who has spent the last well, I guess it's 3 years now, trying to make sense of community profiling data from shallowly excavated soil. So what happens not too long after you dig a hole? And it's been great to actually kind of be involved with this particular journal club again, because it is so open and because I personally want to maintain some sort of literacy in all things microbiological because these systems are so cool. And the paper that we're looking at today exemplifies this. And it's also a nice shift back from I Contain Multitudes, which was our last book, which was all about sort of pulling the picture together of microbes from DNA inference, whereas this paper, which is, as you can tell from the plethora of movies and lovely colored diagrams, moves back into thinking about different genomic configurations, a lot of deletions and deficiencies of systems, really looking at the phenotypes, seeing what's actually happening as a consequence of parts that are lacking with the secretion system that Karen, I suspect, is going to introduce for this. So that's all I have to say.
Okay, thanks, Nicole. All right, so I'm gonna start to get straight into the paper. And so the paper that we're reading, if I'm gonna hold this up and nobody's really gonna be able to see this title, hopefully. It's called Type 6 Secretion System Substrates are Transferred and Reused Among Sister Cells. And in terms of what type secretion systems are, I compared this on the post to kind of like Power Ranger megazords. And the reason why I did that is because they are massive, massive protein complexes that have various different components. And what they do is they assemble together. And once they assemble together, they can start delivering proteins, RNA, different types of chemical compounds, or proteins, DNA, RNA, and different compounds straight across multiple membranes. And there are lots of different types of secretion systems. For this one, type 6 is a double membrane spanning secretion system. And so there are, in terms of classification of secretion systems, there are at least 6 that I know of in gram negatives. And so type 6 is the last one. And I have this neat review here. It's by Costa, 2015. It's a Nature review, but it has this nice figure of all of the different types of secretion systems in gram-negative bacteria. And type 5 is a single membrane spanning, but we won't get into that too much. But at any rate, there's a wide range of secretion systems out there, and they're creatively named by the order of discovery. So just whichever one was discovered first is whatever type that is. And so the one that we're discussing today is type 6. And I don't know if you can see this, but hopefully you can. Can you guys see that?
Let me pop this up really quick. I'm gonna do a really quick thing where I'm gonna try to oh, will it let me share my screen? I don't know if it will or not. Darn it. Because Nicole actually pointed out a really good I can't figure out how to share my screen. I'll work on that. But Nicole actually pointed out a really good review of the type 6 secretion system. So I brought it up. I've got the little figure on my screen. If I can figure out how to share my screen, I will try to do that. But go ahead, sorry.
Okay, all right. At any rate, for the type 6 secretion system, it's kind of like we were talking about in the post. It's very much like a bacteria spear gun. So there's a number of components that come together to form this secretion system. And so I'm not sure if you can see this, but anyway, there's a base plate at the bottom, and there is yes, so there's a base plate at the bottom. There's also a sheath that surrounds the needle complex, and there's a needle complex, and that needle complex essentially shoots through both membranes. And at the end, the tip complex and the tip complex is delivered, and also different effectors are delivered from the bacterial cytosol of one bacteria into the bacteria cytosol of the next bacteria. Is that clear? Oh, I can't hear you, Laura. Oh, there you go! Yay!
Sorry. So yeah. So. This is what I was trying to do. I apologize for taking so long to get that, but the type 6 secretion system that Karen was just talking about, here's a bit of a color graphic that Nicole sent me right before we got on here that kind of gets at what Karen was just talking about. And we've got the different components there. Karen, we've got the sheath that you mentioned, right?
Right.
So
here, I can't actually, it's actually really hard for me to see.
Oh, true, because you're on your phone, sure.
My phone screen. Okay, I'll do my best. But so there's the contractile sheath that's on the outside that I think, by the way that they've described it, kind of squeezes or pushes the tube out. Does that sound right?
Yep. Yep.
And so and then there is the clip. So if I've understood, I haven't read, if I've understood what I've read correctly, is the sheath is made of the two monomers called VIP-A and VIP-B.
Yeah, that's what I have too.
And then there's the there's a clip V ATPase that, what does the clip V ATPase do?
That's the one. That's the one that allows for new sheath assembly.
Right. Okay. Clip V ATPase allows for new sheath assembly, and then the HCP is a hexamer ring that's also shown in, yeah. So it actually, your figure shows the HCP hexamer rings, and then they form. I think it looks like they stack on top of each other to form the tube.
Right. Yeah. I mean, this is really cool. So these HCP hexamers, they're these rings that form this hollow tube. So what Karen's describing, you've got this hollow tube that is assembled, as far as I understand, in the cytosol. And so that's what they're showing kind of in the middle of this figure right here, is the assembly of the tube, but then also the assembly of the sheath that goes around the tube. And so when the sheath contracts, what they're showing here is you actually get kind of like what Karen said, like a spear gun. You actually get the tube kind of ejecting out from the cytosol across the cell envelope and into the target cell, which at some point they describe that in the introduction. They just say, upon an unknown signal from the base plate, the sheath rapidly contracts to about half of its length and ejects the tube with the associated effectors out of the cell and across a target membrane. And I just wrote a note next to that that says, holy crap, with lots of exclamation points, because that's really cool.
I'm going to jump in here and say that I also find that fascinating, because the one thing that, well, there's a couple things that are mentioned, especially with the clip protein that's unassembling or dismantling the VIP-A-VIP-B complex. It is said that it is doing unfolding, or that's how it's described. So to me, this whole thing is rather fascinating, because what I'm intuiting, although I'm not a structural biologist, is to get some of that contractile activity. I'm wondering whether, like, when this thing is built up, you've got a whole bunch of sort of, I guess, intermolecular interaction that leads to a whole bunch of spontaneous conformational change in that protein to allow it to eject that tube, which is fascinating. In addition to the fact that when you think about disassembling it, you also need to be careful enough to not deploy it or make it spring again while you're taking it apart. So there's actually quite a bit of interesting conformational and physiological things at play to put this thing together, deploy it, and take it back apart.
Yeah, this is a lot of like nanoscale physics. This is really neat. This is really neat. That's my really, I was just gonna say, that's my really intellectual contribution to this. This is really neat.
Well, I mean, the thing is that, like, it's for crystallographers, you have to start crystallizing each I just want to put in perspective how difficult it is to kind of understand how these things are put together and to put a complete picture, because you have to crystallize these components, or you have to crystallize them in their individual parts, or you have to crystallize them all together as a whole. And these things have parts. Sometimes they're in the membrane and sometimes they're in the cytosol, but to crystallize all of these components and see how they work together is actually a very difficult process. And the type 6 is particularly unique because there are so many different things that people are discovering from type 6. And so if I'm not entirely sure if the author, Marek Basler, is now in a group in Switzerland, but I read that he also wrote a paper with, I can't remember his first name. Oh, John Michelanos. And he is the big lab in terms of, if I remember, that study type 6. And they did a lot of other neat type 6 papers. So I think that, I don't know if, I'm not quite sure what the relationship is, but there's definitely a lot of good type 6 work that's really neat that's going on here. So not just this paper, but a lot of other type 6 interesting things. Like, for example, there's they talk about how like bacteria use type 6 to attack each other. That's another paper, but that's for a whole other day. But anyway, all right.
Yeah, yeah, no, it's a good system to know about because it's there's a lot of interesting function, and the fact that as they point out in the introduction, it looks like this occurs across a wide range of bacteria. So I mean, this is a significant molecular machine to be thinking about in terms of how bacteria interact with each other, both self and other.
So just on a more, I know we should get to the paper, but I really want to ask Laura this. Just on a general perspective, does type 6, because I know that you studied predatory bacteria, so do secretion systems ever come into play in terms of the way that you think about predator and prey interactions?
Yeah, definitely. And I have to admit that I'm not as up on that literature with secretion systems as I'd like to be. So one of my goals in literature reading is to specifically, as you just pointed out, the different types of secretion systems. I know there are labs that are looking into the molecular biology of those secretion systems in terms of their impact on predation. And so they are significant. And I did, not trying to skip ahead, but there was a certain point where they talk about the cells rounding in the paper, and I'm gonna not have it pointed out now, of course. But there's a part in the paper where they talk about this, I think it's the recipient cells that are rounding. And that was interesting to me because when you have invasion of a prey cell with a predator, you get something called a deleplast, which is a rounded version of your prey cell. So if you start out with a rod-shaped E. coli cell and it gets invaded by a predator, you end up with this rounded deleplast structure. And so I thought, ooh, that's interesting, because that's a parallel kind of change that we'd see with predation. So this is a good thing for us to be thinking about in my lab.
Okay, so then thanks, Laura. I just wanted to ask you that, but
sure.
I guess get on to the paper. So for this particular paper, they are looking at the exchange of different components between sister cells, as the title suggests. And so the first experiment, how the basis of the experiments that they do is, I'm going to try my best to explain this because even for me, this was kind of difficult to grasp. And so we'll kind of work through this together. So they have, they use two different types of strains. They have a donor, and they have a donor type strain and a recipient type strain. And so usually the recipient type strain is type 6 negative. So it doesn't have an intact type 6 system, but it has a GFP reporter of a type 6 component. And then the donor strain has an intact type 6 system that has a red reporter attached to a type 6 component. So recipient is green, but type 6 minus, donor is red, but type 6 positive. And so what they're looking at is they incubate these donor and recipients together in a specific ratio, in this case, 4:1. And so and then they look at the GFP signal in the recipient. So the recipient itself, because it doesn't have an intact type 6 system, has no green signal. So in figure 1A, you'll see that there's no green in any of the mutants, or in any of the, there's no green in any of the mutants, because, so what the outcome is, if you get assembly of a type 6 secretion system, you'll get a green dot on the bacteria. And that's how you know that the type 6 got assembled. And so in the mutants, when in terms of, there's no type 6, so there's no green. But when you mix them together with a donor that has an intact type 6, you get a green dot. So that's what from figure C to G is. So they've deleted different individual components of the type 6 secretion system and then incubated them with this red donor. And so in the first lane of the figures from C to G, you'll see this green and red overlay. So they show that some of the cells that are red, you see red dots because they've assembled type 6. But in the green cells, you don't see anything or in some of them you see some things, and some of them you don't see anything. And so at the so we'll just go through the individual different components. So for C, you have your recipient does not have VGRG. And then when the recipient doesn't have VGRG, it still assembles. So VGRG is the tip of, is the ejector tip complex at the very top of the secretion system. For the next one, you have, oh, there you go. Thank you.
So that's, so we're talking about, I don't know if you can see my arrow. So we're talking about this stuff right here, right? In these panels.
Yeah, I actually can't see your arrow.
Oh, no, okay.
No, these are the correct panels I'm talking about, but I actually can't see the arrow.
Okay, that's not a problem.
Yeah, but at any rate, the first one, the VGRG is part of the tip complex. VIP-A is part of that sheath, and CLIP-V is the assembly, disassembly ATPase that we were talking about.
So let me ask, I'm sorry, I'm going to pause you because I'm going to ask a question because this has been a busy couple of weeks for me. So my brain is not firing on every cylinder that it possibly could be firing on. So I just want to make sure I understand what's going on with the different fluorescence reporters. So we're not looking for, they're going to fluoresce regardless, right? So if the recipient is always going to be green because it is going to be expressing VIP-A. Yes?
Yes. But
and then the donor is going to be red because it's expressing CLPV. So are we looking for an overlay of the two to show that there is assembled type 6 secretion systems? Or are we looking for, I have to admit that that's one of the things that I didn't have time to kind of suss out, was looking for to indicate an assembled type 6 secretion system. Is it an overlay of red and green demonstrating that both of these reporter proteins are present in the assembled type 6 secretion system?
Well, they're only assessing, they don't need to assess the red. They're only assessing the green. So what you're looking at in the black and white panels, those are just the GFP from the recipient.
That's just GFP. Okay.
And
got
the
it.
Reason why, and the reason why they do that is because the donor already has an intact red system. They're trying to see if the components from the donor have been delivered into the recipient. So because the only component from the donor that's red is the CLIP-V.
Got it.
But you don't get an intact type 6 secretion system GFP dot unless the type 6 is assembled.
Okay, so what we're saying is that because this is a component of the type 6 secretion system that's expressing GFP, if it's been assembled, all those proteins are going to be concentrated into one part of the cell envelope. So you're going to see it visualized as a fluorescent, as a highly concentrated fluorescent signal.
Yes.
Whereas otherwise, it's going to be a fluorescent protein in the cell, but it's not, is it just going to be kind of in the cytosol? So it's not going to be showing up unless it gets assembled?
Right. I think I think that you're making a very good distinction, and it's because of some of the assumptions that I already have, which is great. So that's why you see the diffuse GFP signal within the cell. So like they're bright, but there's no concentrated one area that's one dot.
Got it.
So
but if you
sorry, continue.
So that's what panel A is trying to show us, right? Is that you're not in the wild type, you're actually seeing that there is concentrated fluorescence because it's a wild type, so it's going to be able to concentrate those type 6 secretions, that type 6 secretion protein VIP-A, into a machine in the cell envelope where you're going to have a lot of VIP-A, so you're going to see that as a dot. But once they have those mutants, what you can see is that those dots go away, because even though there's a GFP expressing protein, it's not being assembled into a particular point in the cell envelope, so we're not seeing that concentrated signal.
Yes, absolutely.
Got it. Okay, that helps a lot for me, because I was like, my brain last night was like, don't know what this is. I can't figure this out.
Well, even for me, it was that like I'm used to looking at, and I think you bring up a really good point in terms of like what I'm used to looking at, because I don't actually, when we're looking at immunofluorescence, or at least for my lab, we're very used to looking at punctate dots. So if we're talking about diffuse signal, it's kind of like I almost assume and filter it out that it's not really there.
Got it. Got it.
So, but that's a really good point. So that's exactly it. All of the components are concentrated into this assembled type 6 secretion system. So then that's why you get the focused dot of fluorescence.
Okay, and so that's the way they're showing complementation between the donor and the recipient, because if you didn't have the donor there, as they show in panel A, you're not going to get that assembled system.
Yes.
Got it.
Okay. So for panels C to G, they're just showing the different components that they're looking at that are delivered, and they can get assembly. So if you have the tip complex, which is VGRG, so if the recipient does not have tip complex, but the donor does, you still get assembly. If the recipient does not have the HCP hexamers, so the stuff that makes the tube, plus the tip complex, you still get assembly, which probably suggests that the donor is giving the recipient those components so it can assemble. And then so if the recipient doesn't have the HCP alone, then you still get assembly, but they talk about what that looks like in a later figure. If you have if the recipient doesn't have the HCP hexamers plus the tip complex, and the donor doesn't have a sheath protein. So if it doesn't have a sheath protein, it's not going to be able to assemble a type 6 to even deliver into the next one. So then you still end up with diffuse fluorescent signal. And then if the recipient is deficient in the sheath complex, but the donor is not, you don't get assembly, because the sheath in the recipient can't assemble to make an intact type 6.
Got it. Okay. Got it.
That makes sense.
Yep, and that's G. That one you were just talking about was what G shows.
Yes.
Got it. Good.
All right. So in this figure, what they show is that they demonstrate the concepts of their system, and then at the same time they show that the donor is required for this assembly of type 6 if the recipient is deficient in it. So as long as the donor is there and can deliver type 6 intact type 6 components into a recipient, then a type 6 is formed in the recipient.
Got it.
And so they show in H just in the figure, and in H they just quantify how many type 6 you have per cell. And then in panel I, they show the distance that's required. So most of them, you get assembly when the donor and the recipient are very close together. But as soon as they're further apart, you don't see sheath assembly.
One of the things that I thought was really striking about this was, I mean, and that you could see this on the y-axis for H and I, is they're talking about number of sheath assemblies per cell during 5 minutes. And I know these are sheath assemblies, but it's just like the time scale on which the stuff happens in cells is really astonishing if you think about it. You know, like I would have I mean, within 5 minutes, I could maybe build a slightly passable Lego house. Maybe on a good day. But like we have bacterial cells that are assembling these complicated nanomachines. You know, it's really amazing to think about that in terms of the time that it takes. To me, it seems very rapid.
And well, I want to add to that, because that's there's also things that I'm seeing in my experiments that contribute to this. So because of the different types of stimuli that you can have to induce a bacteria change, like different chemical components or even temperature, the time it takes for them to change is actually extremely small. Like in terms of talking about 5 minutes, if you think about the experimental difficulty that your bacteria are going to change from one state into another within 5 minutes, that means that sometimes when you're assessing these stages, it's extremely time sensitive, because as soon as the temperature changes, the bacteria itself also changes.
Yeah,
so,
true.
And so in terms, like I've been having difficulty with this, but in another in another way of looking at it, you know, bacteria are amazing. They can respond incredibly quickly to their environments.
Yeah, it is pretty incredible.
Okay, so in terms of, and so what they conclude from this is there are certain components that are required from the first figure, is that there are certain components that are required for transfer between donor and recipient. And those components in particular are the sheath components have to absolutely be there, but the HCP hexamers and the VGRG tip do not.
Okay, excellent.
I'm just going to add to that, because it is mentioned in the paragraph that goes to with this figure, is that dunking your recipients in just lysates or supernatants does not work. You don't get assembly. You actually do need to have your cells around too. So I just thought I'd bring that up.
Yeah, and that's why they invent, in the end, they conclude that this is very specific. It has to be type 6 delivered, because one has to have a intact type 6 to deliver to the next type 6, because they can't assemble just by components. If you incubate them in just a bunch of components, that doesn't happen.
Yeah, they really did a good job running a number of important controls that were probably, I mean, these controls amount to 1 or 2 sentences, but I'm sure it was quite an effort to run those. And they've done a great job kind of knocking off alternative explanations that people might come up with just through the control that Nicole pointed out. And then also they've got a paragraph talking about, what could it be that it's DNA or RNA transfer? And then they can eliminate that too. They're like, nope, it's the actual protein.
Okay, so moving, is it okay if we move on to figure 2, or is there, do we have
Yeah, sure. Yeah.
things we want to talk about? Okay. So from this, they move on to assessing things that are delivered from the type 6 secretion system. So the things that are delivered into the next cell are the tip complex, so the VGRG, and then this other effector. So effectors are just proteins that get delivered through the tube into the next cell, called Vas-X. And so they, what I think the most, in terms of the highlight of this figure, is they talk about the amount of time that it takes in terms of, so they have the, in B, they talk about the number of number of sheaths per complemented segment during 5 minutes. And that's just looking at those, because when you look at in figure, I think it was D and E. And so if you look at figure 1 in the D and E panel, there's very different types of fluorescent punctate dots. In 1D, the dots are very focused and specific, and there's only 1 per each cell. But in E, you have dots, but they're not very focused, and there's lots of them per cell, and they're much, much smaller. And so that's what they're talking about in terms of they're analyzing this component. They're analyzing this, and then they're looking at how many of those types are also dependent on the presence of the effectors, so the tip complex and also the effector, so that's the Vas-X, and then the tip complex, which is also the VGRG. And so that when you look at the effectors, so things that are delivered across, there's no difference in sheath, there's no difference in the number of sheaths that are assembled. But if you look at the actual sheath components, so the HCP tube, and then if you look at anything that has to do with the actual assembly of the tube, you get more sheaths that are complemented during 5 minutes. If you get rid of, if you have the recipient is absent of HCP,
yeah,
then you get more sheaths that are assembled.
And that's looking at B?
I think so. Yeah, but the no, that yeah, so that's looking at 2B, but that's looking at 1D.
Okay, got it, got it. Yep.
Sorry, 1D and 1E.
And then B is, and 1B is talking, or 2B, I'm sorry, is talking about the number of sheaths per transcomplemented cell during 5 minutes. Is that?
Yes.
Yeah. Got it.
So if you have, so 2B, where it says it has the high HCP, where it's about 3 per cell, that is corresponding to the individual diffuse state dots in 1E.
Got it.
Does that make sense?
Mm-hmm. Yeah.
Okay.
Yeah, and I think they've done a good job. They did a really good job with their headers and their figure title, their figure captions too, which is not always the case. I think that's a hard thing to write, but they've done like in figure 2, they're like tip protein concentration dictates number of sheaths per cell, whereas HCP determines sheath length. You're like, oh, okay, that's that makes sense.
Yeah. So the reason why it's if you're watching and it seems very confusing to follow along, I think it's because like there is a lot of good science and detail that's gone into this paper, but it takes a while to process and to connect together.
Yeah, definitely. But I mean, this is an intense amount of work, so it's they really built their case very well.
So what I'll do is I'll actually just skip to I think what is the best part of figure 2, and I think that's probably the way that we'll do the rest of the paper is I'll just skip to the part where I think is the best.
Yeah, that makes sense.
If that's
Yeah, that's fine.
okay. In terms of that illustrates the best point, which is 2D and 2E. And so what they do is they complement the tip, and then they complement the HCP on a plasmid, and it's inducible. So in terms of being inducible, it means that the concentration of this protein is going up over time as soon as they induce it. So at time 0, it might be very, very low. And then as they induce it with, I think they did IPTG. So as they induce it, the concentration of this protein goes up. And so as the concentration of the protein goes up, you see more and more assembly happening over time. And so that's why they've made this conclusion that it's proportional to the concentration of these components, because as they're expressing the components more and more over time, you get more and more assembly.
Yeah, that makes sense. So what they're looking at is if you give it more of the tip protein, so if you induce the plasmid to express more of the tip protein, they're showing for panel 2D, the right-hand side is showing us more assembled type C secretion system machines in that pack of cells than in the pack of cells from earlier. So that's what's showing them number of sheaths per cell is impacted by increasing the tip protein amount. And then 2E, I think their conclusion is that we're looking at, in the one on the left-hand side, like you were saying, Karen, that was a good way to describe it. They're kind of like these little punctate fluorescent dots. But then if you give it more of the sheath protein, which is HCP is the tube protein, I'm sorry. Yeah, HCP is the tube protein, the shaft protein. And so if you give it more of that, then you're going to get a longer sheath length, because you could see that the fluorescent dots are getting longer with an increasing tube protein concentration. Yeah?
Yes.
Woohoo!
Does that make sense now?
Yeah, it does. Yeah, it does. No, you're going through this very well.
Okay. All right. In terms of, let's see if there's anything I want to highlight about figure 3. Efficiently exchanged. I think it's just for time's sake. Like I'm trying to get through this more quickly. But is there let me see, what are the best highlights that I think are the best parts in the paper? Okay, I'm gonna skip to figure 4. And the reason why I want to highlight figure 4 is I think they've developed this very, very unique system for experimenting and testing lysis. And so what they do is they have a a LacZ positive reporter and donor. And so what this, so this reporter and donor produces beta-galactosidase. And so then they put this component in the media called CPRG, and CPRG is broken down by beta-galactosidase, and when it does, it creates the red color.
So we're looking.
That's what you're seeing be.
Sorry, I don't mean to interrupt. So we're looking at the secretion of LacZ into the media, which is going to break down the CPRG, which is going to turn it pink to tell you that beta-galactosidase has been not only produced but also secreted.
Right, but what they're not doing this in terms of testing secretion. They're doing this in testing lysis. Oh,
lysis. Okay, sir. Yep. Thank you.
Right. So I think if I've read this correctly, I'm not. So this is where where I got confused. So I'm not sure if the LacZ reporter cells are E. coli or they're Vibrio. But I'm not sure because they were testing lysis using E. coli. Wait, hold on.
So it says. So there's a bit that says to estimate the detection limit of the CPRG-based assay, we tested lysis of the LacZ positive E. coli and the Vibrio cholerae reporter. Wow, that was very Massachusetts. Reporter. By Vibrio cholerae lacking TSSE. I got distracted by my sudden New England accent there. The reporter.
And at any rate, so what's happening is that as soon as if the LacZ positive bacteria is lysed, beta-galactosidase is released into the media, and the color turns pink. So I thought that was really, really cool in terms of the way that they're testing it.
Mm-hmm. Yeah.
And so what they find is that you only get lysis if the recipients they find they only get lysis if the recipients if they don't have I'll word it the other way. They find that you don't get lysis if the recipient is deficient for the sheath protein or if it's deficient for this effector, this TSEL.
Right. And you can see that because of B shows their 96 well plate. They've got them all in a column there, right? And so the two you just mentioned are still kind of that orangish color.
Yes.
Got it.
So, I just wanted to point this out because I think it was a really, really elegant system in the way that they've designed this. And they can test multiple factors because they can manipulate the genetic elements of the donor, the recipient, and of the reporter.
Yep.
Does that make sense? Okay.
Yeah, and there's... Go ahead, Nicole.
Oh, sorry. And I think the other thing, which, it's going to be obvious to Karen because of her familiarity with this, but the other thing that's nice about doing a lysis-based reporter assay is it's proving the equivalency of efficacy of these reassembled and complemented secretion systems. Like you manage to get through both membranes, regardless of the configurations that allow you to get signal. So that's pretty compelling evidence. Again, yeah.
Can you, for me, can you, Nicole, can you clarify what you mean by that? I think I see where you're going, but I'm a little behind both of you. So maybe.
Yeah, it's sort of a simple statement, but it's just like it's unambiguous evidence that the reassembly of this TSS system in your type 6, like you're making it something that's capable of busting a cell open, kind of thing. We don't really see the LacZ conversion until that LacZ's in the media, and you have to break your cell open to get it. So structural efficacy of the speargun doing the speargun thing, even though your recipients aren't making out the speargun all on their own.
Got it.
Yeah.
Got it. Okay. Yep. That makes sense.
Right. And so I'm sorry, I didn't actually illustrate the most important point of this, which was to, as an, it's an assay to test whether or not they have a functional type 6 that's transferred. So it's one thing to look at whether or not you get that small green punctate dot, so it's assembled, but you don't actually know if it's functional. And so by doing this, they can show that this type 6 secretion system that's transferred from one cell to another is functional because it actually lyses
Right.
bacteria.
Right.
Yeah.
That makes sense. It's cool.
All right. Is there any other part? I know because we're short on time. Is there any particular figure that you guys wanted to talk about?
Well, we've got 5 and 6 left, and then 7 is their model. So my thinking is we should probably sum up with the model, but I don't know, was there a point that you wanted to make with 5 or 6, Karen? I mean, you've done a great job walking through this. I understand this a lot better than I did yesterday when I tried to read it.
Oh, thank you. I mean, I'm sure that there's definitely more things that I could said, or I could have said things in a lot less words, which would have taken a lot less time. But I guess so I guess there was only one the one other experiment that I really appreciated was the one they incubated the bacteria with chloramphenicol, antibiotic that prevents protein synthesis in wild type cells, and then they incubated it with donor cells, and then they noticed that the donor cells were still able to transfer into components into wild type cells, and those wild type cells still assembled after chloramphenicol treatment. So I thought that was really cool because after the assembly is they've inhibited protein synthesis, so the wild type can't make any, but as long as there's a donor cell there that can still give them some, then that's it can still assemble, and I thought that was really cool.
And so this kind of starts to get at the broader picture of this. Like, what do we think is the what's the larger impact of this in understanding bacterial, there's 2 ways to think about this, is that your sister cells, as they call them in the title, are potentially cooperating with each other. I'm teaching symbiosis right now, so we're going to talk about this way. Your sister cells are cooperating with each other potentially by sharing these components of the type 6 secretion system. But then they also start to bring into it the idea that you've got conflict introduced because these cells are now going to make sure that they can attack or compete with other cells. So, I mean, they're using Vibrio cholera for this. I'm curious, like, Karen, what do you think about where this is going in terms of understanding things like virulence, or understanding things like how bacteria compete with each other in nature?
How bacteria compete with each other. So are we talking about interspecies, or are we talking about intraspecies?
In this case, I would say we're probably talking about interspecies, because my guess is that within a clonal population, at least, they're going to be those cells are going to be sharing the type 6 secretion system. Although, I mean, did they get much into like how much genetic distance is needed before you stop sharing?
No,
type 6?
No, I don't think so.
That would be interesting.
It's the same Vibrio background, but that's a really interesting question.
Yeah, that just occurred to me. That would be kind of interesting to figure out, like, how much sharing happens, because they all yeah.
So I mean, they do talk about a little bit in the paper in terms of they try to talk about how they have a proof of concept that in polymicrobial community cooperation between 2 strains based on type 6 dependent protein exchange could contribute to killing of a 3rd strain.
Got it.
So
yeah.
You were talking about clonal cooperativity between this once, or clonal so you would get clonal cooperation, but you might even get cooperation between 2 slightly different clonal species, but they might contribute to the killing of 3rd strain, for example, E. coli.
Right. So let me just make sure, because if I misunderstood this, I want to make sure I have this. In all of this experiments that they did they ever set up an experiment where the sharing was done between 2 different species, or was it always one type, one strain, and then a mutant of that strain, and then they might introduce like an E. coli or a different strain in that assay with the three, where there was the donor recipient reporter.
So I mean, they did an experiment with P. aeruginosa, but I think that was P. aeruginosa exclusive. I don't think any was separate, like a Vibrio plus.
Okay.
P. aeruginosa.
Got it.
Because I think what they were trying to show is that that you can also get transfer between P. aeruginosa Pseudomonas cells, but I don't think they did. And correct me if I'm wrong. I don't think they did one like a Pseudomonas plus a Vibrio.
I don't think so either, as I remember reading it.
All right. But yeah, Nicole, did you have any other comments or thoughts?
A couple. So, I mean, getting back to this idea of cooperation. So, this is going to be a bit roundabout, but I'm trying to imagine that I was a postdoc who was told, create a recombinant expression system that makes this entire type 3 system in like some non-heterologous bacteria. And I can't imagine making anything that would either not do it, spit it out as inclusion bodies, or make cells that really hated being alive and looked really pathetic and didn't grow well. So there is a lot of crap. This thing is huge. We can see it from that fluorescent signal, like in these microscopy experiments. And I think that's sort of thinking about that, like offsetting your metabolic load to maybe just a few cells in this Vibrio population who are going to do the heavy lifting of building all these parts and sharing it makes sense energetically, particularly in a pathogenesis context where you may actually have to go most of your community in the process of killing off people, like not people, bacteria and people eventually, to get your foothold in wherever you are. You're going to be up against innate defense systems, other chemical methods of trying to make sure that Vibrio doesn't end up the winner. So like there's all this other metabolic crap that Vibrio has to do in addition to making these spears. So it's kind of an interesting way of disseminating biochemical responsibility. We're just going to pass these syringes around so that maybe you can devote the rest of your anabolic repertoire to like making other things you need to do to make sure you win the war and hold down your turf, which is kind of, I know it's an extended metaphor, but like I think thinking about it that way, this is fascinating. The other part that I thought was really cool is, I mean, I never did the whole organismal biology thing, so like there's nothing but jealousy when you've got like a system where you could actually say, yo, this syringe is shorter than this syringe because of like, you know, we didn't introduce the tip complex and we only get really, really stumpy ones when we put the rings in. And that to me is also fascinating because part of me is starting to wonder like how much dependency on assembly and stabilization of protein structure do you need? Like, what is the order that this thing gets put together? So like, is the tip there because it actually locks down wavy parts of your ring protein when they start stacking on? And is that why like the HCP complementations are so much shorter? Because there's a whole bunch of other, I'm going to say they're chain reaction-y types of things to lock that you know, that supramolecular structure into place. So there's some really awesome biochemistry questions that come out of this in terms, and also the questions about what is the sort of social strategy between passing around your secretion system parts.
Yeah, definitely. That's all true. Yeah.
Oh, I really like that because I want to add on to, I want to, like when you were talking about whether or not they're sharing needles, like that also brings up the question whether there are cheaters in the population or whether a single population actually divides up the number of people who produce type 6 secretion systems because they can just share among each other. So if there is only a proportion of the population that produces it, then there's like you can have a minimal number producing and still get the same effect, and then everybody else has another metabolic role within community compared to like compared to just having everybody produce type 6 at once. So I think that that would also be really neat to study in terms of like, are there signals between bacteria that say, okay, you make a type 6, or I'm making a type 6, you don't make one? Or so going back to Laura's question earlier, what is the broader sense of, in terms of virulence? Like, you can get inter-community bacterial cooperativity because you, if you have this sharing, then metabolically you can save energy and do other things in terms of like that benefit the community as a whole, and then you ensure the survival of lots of bacteria. I think the downside of, in terms of studying virulence and thinking about this, is in virulence we're very used to thinking about the bacteria as an individual. So it's like, okay, one bacteria invades one cell because initially the population is so small in terms of inoculation population. Like, you never think of the bacteria acting cooperatively as a whole. And so, I mean, in terms of metabolic trade-offs or things that happen in community, that wouldn't even happen until much later in the infection, when you get more bacteria that are able to do these and share these responsibilities. And I think in terms of the future of virulence, that's probably where we should be going. We should be trying to understand how these, instead of looking at it from, okay, from a one bacteria perspective, which is useful in initial stages, but rather looking at it, what happens in later stages of virulence when you have lots of populations, and how do these populations cooperate metabolically, and how does this affect virulence as a whole for the population, rather than in the bacteria population, rather than for one particular bacteria. Or another way that we assess virulence is, does the host die? So those are very clear black and white outcomes, and I think we need to start to move away from that and try and decipher out the individual nuances of infection. Anyway, okay, and I think that comes to the end of our time. So does anyone have any last questions or comments?
I don't think so. Karen, thank you very much. I'm really glad that you led us on this one, because I think this was a great paper to read, and then I think you explained it really well. So I'm glad I read this, because I'd seen things about type 6 secretion systems, but I honestly didn't know too much. So now I'm a little more up to date on what that is, and that helps us for our own stuff, as you asked at the beginning. So
I just want to give a shout out for Joel, who suggested that we read this paper in the first place, but he's not here, which is unfortunate. But so, I mean, thank you, Joel, for suggesting the paper, and I hope you get to watch this, and I hope it clarifies things for you. Or if you have any other comments, please tweet us. So if anybody else is watching, also, if you have any comments or questions, you can tweet Laura at microsci, or tweet Nicole at Old Dusty, and you can also tweet me at Karen, spelled with a Y, Low, and we'll post all that information on the website and also on YouTube. So thank you for watching, thank you for joining us, and we hope to see you next time. All right.
bye.